So the first session that we have is engineering and new technologies in neuroscience. So finding innovative ways to combine rare data, multiple methods to make new discoveries. And the first person that I wanted to invite in is Dr. Satrajit Ghosh, who is based here at the McGovern Institute and at Harvard. And his research interests with his lab are using machine learning for precision psychiatry and medicine, trying to understand the neural basis and translational applications of human spoken communication, and also being an advocate for reproducible research and knowledge generation. So come on over.
I haven't been feeling too well over the last week, so if my voice or other things go, just bear with me a little bit. Thank you very much to the Polonium Foundation for inviting me to this. It's an incredible talk to follow after Anna. So I'm going to try my best to kind of tell you why some of the ways we are thinking about science kind of perfectly maybe reflects what Anna was talking about in the previous talk. Just to give a thank you to our group members and many other collaborators and projects that we have worked with over the years. I wanted to put this up there before kind of going through the rest of it. My own journey started as a computer scientist, and then I became a computational neuroscientist. And my computational neuroscience work was focused on speech motor control. Hopefully this won't keep happening, or I hope. I'm not pressing something, but and to me, this was an interesting system. Most of us speak most of the time, and we communicate with each other. And when we speak, our own brains are listening to our speech. That's how we acquire speech. And other people are listening to our speech and giving us feedback. We take that feedback. So there's a very nice closed loop system set up. And closed loops are important. And closed loops allow us to learn in various ways. And I want to keep this thought process in mind. And partly, I'll pick up on a statement made earlier today, from brain drain to brain circulation. I'll take that and say from information drain to information circulation. I'm going to come back to that notion of how we need to keep information flowing constantly across the ecosystem over time. But it's not just this closed loop of taking actions, measuring, and adjusting. We also build models of what our world looks like in our brain. So we are also constantly asking our brain, we think, is asking, what will happen when I take this action? And it takes that notion of what's going to happen together with what actually is happening and uses those signals to learn. So this has become a system that we have studied for a long, long time. What we do today, as was mentioned, we try to understand the neurophysiology and translational applications of communication. Communication breaks down for various reasons. The second thing we do is use machine learning for AI and for personalized medicine, and developing interventions that tackle this closed loop problem. And finally, we use neuroinformatics as a way to connect information across many different pieces of data and knowledge. Okay. I'm not a psychiatrist, I'm not a clinician. I'm a computer scientist and a computational neuroscientist. And I accidentally came into this world of mental health when some colleagues asked me to help out with trying to say, will this person benefit when I give them this drug? And why is that important? Even today, and this was written back in 2012, but even today, the first time a drug works for something like depression is about 30% of the time. And that hasn't changed a whole lot. So if we can, again, shift that needle a little bit through understanding how we get information about an individual into the space, it would be really nice to be able to move things forward. So mental health has been driving some of the considerations, but it is challenging. And why is it challenging? Because when we look at the landscape of human neuroscience, this is kind of in contrast to Anna's space. She's starting at the molecular level and going up. We started at the human level and asked the question, what can we do at the human level with the tools that we have? And it's hard because it covers many different elements that come. I'm just going to go through a few of these things. Just at the top level, we are thinking about neurobiology. We are thinking about context, environments, and social determinants. We're thinking about timeline, aging, lifespan, behavior. When I'm speaking, that's a different timeline of behavior than when I'm memorizing things or have memories of things. There are clinical aspects, obviously. And then when we think about analysis or modeling, we are approaching this with many different tools because we have impoverished information. None of it is perfect. I think it was funny when Anna said, we can go down to 7 angstroms, but we need to go further. That's what we do as scientists. We want to peer into things that we cannot see. The Hubble let us peer into the world in different ways. Our microscopes do so. And so what we have been doing is kind of working in this space of trying to integrate information. And I'll give a quick, this is the only scientific slide I'm going to show from our group. This is work going on with Sohei Kim at UMass Medical. And she's imaging 6-month-old infants while they're in the scanner. That itself is a challenging problem while they're sleeping. And she's asking the question, do they have a preference for their mother's voice? That's what we do as scientists. We have a question, we want to answer it. And do we have the tool to answer that question? When she started this, we didn't even know if the brain was going to respond to sound while an infant is sleeping at 6 months of age. That knowledge wasn't there. So the first thing was, will it respond to voice over other kinds of sounds while they're in the scanner? And yes, if you're looking at a brain and if you think of the colors as the brain responding to things, the brain does respond to mother's voices. But this is any mother's voice. We presented all kinds of mother's voices. Then the question was, does it respond preferentially to the kid's own mother voice? And there are very specific areas of the brain that are involved in social cognition that show a preferential response to the infant's own mother's voice over other mothers' voices. Finally, that response to their own mother's voice is related to maternal postpartum depression. It's related to maternal postpartum anxiety and infant negative emotionality, which is an estimate of emotionality in the infant. So this is just, I think, giving an example of the set of things that when we are looking at human neuroscience, it goes into a place. We did not know the answers to any of these questions. That's why we need to continue to both ask questions and get at experiments that can answer those questions. All right. This was on new and emerging technologies. None of the technologies I showed you earlier were new or emerging, right? It's about fMRI in a scanner. We've been routinely doing it for about 30 plus years. In fact, the Boston area was one of the places that co-developed functional MRI. But we are looking now at the US Brain Initiative, which has been supporting the development of many new technologies across cells, circuits, and behavior. And we're kind of lucky to have funding from a variety of resources to kind of investigate this space. So one of the things we've been doing in this space is making information generated by these initiatives available to the wider world. And towards this, we've built something called the Dandy Archive, which houses a certain type of data, neurophysiology data and some proteomic imaging data at single cell, at whole brain level in humans. We're distributing about a petabyte of data right now to the world. Now, when we think about a petabyte of data, most of us don't know what that is. Most of us deal with maybe kilobytes, megabytes, gigabytes. A typical movie is about a few gigabytes today. So that's a lot of data being distributed. Now, is it useful data? That's a question we want to answer. This has been growing, and this is coming across many different species. So it's not just humans. And we are distributing it, as one can see, to almost all the world. It's really nice to see a lot of use even in places like Africa, which used to have much reduced usage even a few years ago. This space is expanding. So we're in a great place to kind of say data itself, or the ability to access data, is a new technology that didn't quite exist at scale, at the distributional scale that we can deal with today. The second thing, and Tomasz is here, and he's part of it, so you can ask him all the questions later on, of a project that's called BICAN. And within this, people are trying to figure out cell types. So Anna mentioned some of the cells she's targeting in some of her work. Here, the consortium is looking at cell types across the brain at single cell level, across species, across lifespan. And that is a very challenging problem. Being able to take something that you can do in a slice or in a sample and being able to scale that up across the brain is incredible. And this consortium has put together a set of wonderful packages that are exploring this space, and all of this data are being made available through various resources. Now, as part of this, there's a lot of knowledge being generated. And one of our roles in this consortium is to aggregate this knowledge, to put it together for access by others when they do so. So coming back to that information circulation theme, we want to be able to make sure that it's not just stays in a lab. We want to get it out there as much as possible to everybody's spaces. In a second consortium, we are part of a project that is trying to understand how are different parts of our brain connected to each other. And here, there are new technologies being developed. One of them in particular, it's called HIP-CT, which allows us to look at a brain, an intact brain, almost close to cellular resolution. We're not quite there at cellular resolution. Using a technique, using an X-ray beam, and there's only one place in the world in Grenoble, in France, which can do this. And so brains are sent over there and they're imaged. But this notion that we want to get at long-range connectivity, we currently don't have techniques that give us precision on how different parts of our brain are connected to each other. And this project, again, is trying to get at and validate this across non-human primates and primates, with the hope that this will get distributed. But there's a further clinical goal. One of the places where this kind of information becomes very handy is when people are doing deep brain stimulation for something like Parkinson in the striatal or the caudate. Where that goes in is a bit of a trial and error by clinicians, and they jiggle it around and they get effects, and then they get to say, hey, this is the place where we put it. Can we improve precision? So a lot of our scientific processes are around how we improve the precision of something. The final consortium project that we're part of is called Brain Behavior Quantification and Synchronization. And this gets to behavior. How do neural circuits give rise to behavior, and could be monitored? So one of the projects in this, and this is made up of 24 different awards at this point in time, that looks at the connections between neural firings and behavior across species. I'll take an example of a human project where somebody's coming in typically for epilepsy. So they're putting an they're putting an electrode in the hippocampal area to reduce epileptic seizures. But that also does recording, because within the system, they're using this kind of closed loop to say when to stimulate. So that recording part can be used to say what's going on in that part of the brain while this person is navigating the world around them. And they're collecting data where they're looking at what this person is looking at, what they're listening to, who they're interacting with. And that kind of information simply did not exist with kind of neural and behavioral recordings of a human. So the hope is we're getting neural activity, we're getting real-world sensors capturing behavior out there, we're capturing behavior in a lab, which is where most of science is done, but not outside. And now, can we get at internal states in the brain that are giving rise to these different kinds of behavior? So kind of going back to my early thing on why I studied speech motor control. We did not have these things when I studied speech motor control. But today, we can start to ask these questions. And even then, I can't study that circuit easily. There's some work going on in some labs that look at some of these surfaces. In fact, in Tomas's home institution at UCSF, there's work being going on with electrode recordings on the surface, but not necessarily in some of the deeper structures, not simultaneously across these. So we're very far away from being able to monitor a system. We have thousands of sensors in a car we drive. We are not able to do that in a human brain at this point in time, in a live human brain. Okay, and together with this consortium, there's a new repository we put out there that will capture data from across these species, including private data from individuals that comes into this place that will be distributed through various access controls. Okay, it's not easy. So just our data coordinating center involves a lot of people. And I wanted to put this slide up there in that science is moving a lot towards team science. The fact that, as you heard from Anna, there are entire consortia or preclinical consortia put together. We need to be able to connect the dots across humans who are interested in solving many of these spaces. And in particular, I want to point out that we have an ethical advisory board and a scientific and technical advisory board because we are getting into these complex spaces. That person who's moving around, we have consent to their information. We don't have consent from all the people they're watching while they're moving around. They are unknowingly part of a scientific experiment that somebody is doing. How do we think about information as we do so in this space? As a result of these consortia, across 5 or more different brain repositories, data are growing at an exponential rate. And yet, most people don't know this. Most people still take their ideas and start an experiment in their lab. I would like that to change. The moment you think of an idea, could you go out there and say, does there exist data where we can start from? Now that data exists, people are building AI tools. Here's a tool where you can chat with MRI or brain images and ask the system to compute things of interest to it. And this was developed here at MIT from colleagues across the street. We're building similar tools to talk to data in our repositories. We can talk and ask the questions about the data, and that has changed the way we are interacting with information. This has gone further. We're asking AI tools to itself build ways of analyzing the data. So we're asking it, can you tell us what we should be looking at in the data? So we are entering a world where our interaction with information is changing. Now, these tools don't have the knowledge that Anna has in her head. And one of the key questions is, how do we get that human-AI interaction to kind of come together towards better science? These tools are also not perfect. We have been hearing about biases in AI technologies across many different sources of information. And as Anna said, Anna talked about, if you put bad data in, you get bad data out. But even if you put good data in, they may be biased in other ways. They may reflect only a slice of the population. They may not apply to other people. So being able to understand what data we have, what data are we missing to improve representation is critical. So we are at a very strange time right this minute, but that's what makes it exciting. We can shape the field in ways to do so. So we are dealing with availability, discoverability, and usability issues around all of these pieces of information. We've done that, and we've done that as scientists. We try out things. But could we scale that up? And that's the question. Could we go beyond our labs? Could we connect, hey, this didn't work in our lab. Now that takes a conference, or these days perhaps a virtual Zoom meeting call, for somebody to convey that piece of information to some other person. So information circulation is slow in science. And this is where I'd like to propose a new way of thinking about how we do science. So I'm going to focus on a little bit of the challenges out there. The complexity of neuroscientific inquiry. I don't think I have to tell you here, our brains are complex. We interact with a lot of things. And Anna focused on some very small parts of neuroscience. To integrate across those things is complex. The data explosion, I've just showed you how we are expanding access to data. But what I haven't showed you is it useful data? So just because we have data doesn't mean it's useful. How do we think about that? There's a lot of fragmentation and lack of coordination. It's a hard problem. So we kind of do this in stochastic ways the way we have done. It's kind of how we did it in villages, how we did it in cities. We're just kind of expanding that as a scientific process. The skill and knowledge gaps persist across the ecosystem. There are things I don't know, but I know that 2 doors down, somebody knows. But we don't know what we don't know easily enough. Could we change that? There's infrastructure inequities. I'm here at an institution that has a robust infrastructure. We can do things over here that many institutions cannot work towards it. Could we make that accessible to more people around the world? Because if we have to solve this human brain, it's a global problem, really. Everybody who can contribute to it should be able to contribute to it. There are ethical and regulatory fragmentation in terms of how data can be shared. For those who live in Europe, just GDPR, which governs some of the rules of regulations of how this is done. Biased attribution, there are many people contributing to this. And yet we see certain papers and certain things as our current attribution metrics. At least in neuroscience, we haven't quite solved the attribution problem. I think it's a little better in domains like physics, where they have been doing some of these collaborative things for a long, long time. And finally, I would say infrastructure is underfunded. We don't do things as a function of scale of a project, right? We don't say take 0.2% of the award and put it towards infrastructure. We also develop infrastructure quite haphazardly across projects, so not in a coordinated manner. CERN, for example, as far as I know, gets a certain percentage of a GDP towards building of that infrastructure and maintenance of it. Could we change science that way to be able to do that? I think it would change a lot of the things that we do. I'm not going to go into the details of this. I've been given maybe now the 10-second warning. I want to see this through a lens of a contract, that when we build infrastructure, we agree to certain things. So this does not mean you can't build infrastructure. This means that we need to be able to have access to things programmatically and as humans. We need to be able to understand how the data came to be, because a lot of the risks is when we don't know the answer to that question. And finally, we want it to be where information in any part of this infrastructure quickly travels to another part of the infrastructure, and that these infrastructures are self-healing. None of these exist right this point about infrastructure. So could an infrastructure be a new instrument for science? And I think it can be. So I'm going to end back with what our group hopes to do for mental health, is synthesize this information through the lens of AI technologies in partnership with clinicians, patients, caregivers, and bring it together. Now, we have worked on many pieces of this, but this as a whole does not exist. And I hope we can build that kind of a system where everybody can participate in solving those problems. In our case, we are guided by the mental health problem, but this particular thing could be applied to many different kinds of problems. With that, I thank you for your attention, and I'm going to pass it on. Hope it wasn't too far beyond that.
Do we have any questions?
Hey, Satrajit, this was great. And my question is along the lines of sort of what makes us human. And, I've always been fascinated by this observation from, monozygotic and dizygotic twins, that monozygotic twins almost always, even if they were reared apart in different parts of the world and grew up in different political systems, still share so many of their personality features that are unique to them and presumably therefore genetically determined. So how far do you think we are from capturing the sort of architecture of human personalities and the underlying genetic and medical features as it relates to the susceptibility to some of these disorders, right? Some of us are more neurotic, others are more agreeable, and others are more emphatic. And I'm just curious how you're thinking about this.
Great question, Tomasz. I happen to know a pair of monozygotic twins, and I can say that they did not get reared in different parts of the world. And they are quite similar in certain ways, and yet different in many ways as well. And in fact, I could say, because I have their brain scans too, that their brains are different. So there's folding patterns are different. You would kind of, and I think this is one of these things where this interaction between genes and environments really kind of comes in. Is personality encoded in genes or that interaction? I will lean towards the latter. I don't think it's encoded fully in the genes. But also our definitions of what personality is a fuzzy one. As I'm not a psychologist, I should point that out, but psychologists have created nice theories and factors, the big 5 factors, right? You mentioned them, as ways of understanding or compressing information. And I think that's where I see data as a different way of thinking about information. We categorize things to be able to convey things to others more easily. But most of the time, things lie on a spectrum somewhere. And these different dimensions that can get exposed, the genomes, the interactions, and why we need systems that can collect information across those individuals over time. Because the genetic blueprint gives us one way of thinking about this information. And again, with the BICAN project, which you're part of, we know that gene expression itself changes at different stages in time, and yet some parts of it remain consistent. So we're scratching the surface on, I think, relating that personality question to what we can see in the biology of things. And we're very far away from that perspective. Now, in a kind of related to that question, one of the things we've been doing is we're analyzing voice and language in our group in relation to suicidal ideation, prediction of disorders that will come up. So we've shown that you can get some information about people to say if they will get bipolar disorder in the next 10 years. Now, there are many ethical questions that come about around this. I'm going to separate those out and say, we don't know what to do with those kinds of models. In fact, I don't know what to do with most prediction models we build. It's one thing to be able to predict some of these things, or use that prediction to understand what information went into that prediction, so that could tell us a few things like, oh, personality may be represented in these circuits of the brain. And people have been doing that for a while. But this is the space where, A, you want to understand, is personality hard-coded or changeable? It reminds me of a fun project that we were funded to do about a decade and a half back. Can you change intelligence? Can you shift the needle on intelligence? Now, what we could say is that one of our conclusions from that project was, maybe we can't shift intelligence, but we know people can become experts at something very well. So we did these studies where people would play these games where they became incredibly efficient at those games. And you would think some underlying substrate would carry over to another game, which requires same skills. Nope, it didn't. And so the generalizability and what the brain is and how it wires itself is still a mystery in many, many ways. And I think one of the clinical cases where this comes up often is PTSD, where people are trying to understand how we think about memories in the context of trauma, and how is it encoded in the brain. So I would say, I know we started with the word personality, and I went into these, but that's what I think personality is. It traverses all of those different